Good afternoon. Today we are going to discuss urinary histology, which is actually the last of the series for ANP2. Now, in urinary histology, we are going to be looking at the renal cortex, the renal medulla, glomerulus, the proximal and distal convoluted tubules, as well as the collecting duct, the ureters, and the urinary bladder. Let's start with the renal cortex. And the most distinguishing feature that you see with the renal cortex is the presence of these round structures which look actually like little flowers. And these are the glomeruli. A single one is called a glomerulus. And so you can see the glomeruli up there, and you can see how they look, at a glance they look like little flowers. And they are surrounded by the Bowman's capsule. So here you're looking at the Bowman's capsule which surrounds these glomeruli. And along with the glomerulus and the Bowman's capsule, which collectively are known as the renal corpuscle, you find the convoluted tubules. You find the proximal convoluted tubules which stain darker, also tend to have the epithelial lining is a little fringed. And then with them you have the distal convoluted tubules, which normally take a lighter stain, tend to have a larger lumen, with the epithelium being not as high or not as tall. Apart from these, you may also notice collecting ducts, and sometimes it gets a little difficult to distinguish between the distal convoluted tubules and the collecting ducts. If the collecting ducts are cut longitudinally, you'll see many of them together, and then that makes it a little easy to identify them. Now let's look at the glomerulus in a little more detail. And here what you see is, you see a capillary network. And so the entire glomerulus is really a capillary network which is fed in by the afferent arteriole, and the exit is through the efferent arteriole. This glomerulus, these capillaries are lined by simple squamous epithelium naturally, so that filtration can occur well. And the glomerulus sits inside the Bowman's capsule. Now the Bowman's capsule has a parietal layer which is on the outside, and we have a visceral layer which is in intimate contact with the glomerular capillaries and actually is part of the filtration membrane. And in between the parietal and the visceral layer, actually you have the capsular space, or what is also called the Bowman space, which is where the filtrate first enters. Now moving on to the proximal and distal convoluted tubules, let's first look at the proximal convoluted tubule. And if you notice in both images, you will see that the lumen, the lining which surrounds the lumen, is a fringed lining, and this is known as a brush border. In the upper image you can see that there is only one proximal convoluted tubule, whereas in the lower image you can see that there are more proximal convoluted tubules. Notice how darkly they stain, notice how the epithelium is fringed, and compare this to the distal convoluted tubule, where you can see a wider lumen. The epithelium is not as tall, it's simple cuboidal epithelium, there's none of that fringed appearance. And in the lower picture, which is very typical, you can see that the stain is, it's lighter stained. So this is a feature of the distal convoluted tubule. Some of the other things that we see, as I mentioned earlier, is of course the larger lumen that you're looking at. Let's move on to the renal medulla, where we are going to look at collecting ducts. The collecting ducts are present both in the cortex and the medulla. Now when you look at the cortex, the identifying feature is the glomerulus. I mean, you look at the glomerulus, and then of course you see the proximal and distal convoluted tubules. You may also see collecting ducts there. When you look into the renal medulla, you will not see any glomeruli, you will not see any proximal or distal convoluted tubules, but you'll see a whole heap of collecting ducts, and along with them you will see the loops of Henle. So those will tell you that this is the renal medulla. Now if you look at the upper image, you can see the collecting ducts cut in a longitudinal fashion, and in the lower image the collecting ducts are cut transversely. In other words, they're cut across. In both, the identifying feature is that the epithelium stains light and doesn't take a dark stain. The lumen is rather large, and along with these collecting ducts, in the top image you're looking at the thin segments of the loop of Henle where the lining is simple squamous, and in the lower image you're looking at the thick segments of the loop of Henle, and these are very similar to the distal convoluted tubule. Now you could mistake it for the distal convoluted tubule, but you notice that in both these images you do not see any glomeruli, you don't see any proximal convoluted tubule, so you would identify this as the renal medulla. You will also see a lot of capillaries, and the difference you can see in the top image between a capillary and the thin segment is that the capillary is full of blood cells. So, don't mistake a capillary for a thin segment of the loop of Henle. Now let's look at those structures which are responsible for transport and storage, namely the ureter and the urinary bladder. Now both these structures are lined by transitional epithelium, and transitional epithelium is identified by the fact that it has many layers, so it is a stratified epithelium. The top layer of cells, which are known as umbrella cells, they have a coating on them which is called the cuticle, and on the top picture you can see the cuticle quite well, and in the lower picture the arrow points to these umbrella cells, which you can see are very large, so hence you can differentiate them from stratified squamous epithelium, because in stratified squamous epithelium the cells are flattened out, whereas here you can see that the cells are large and plump. The arrow in the lower picture is pointing to the basement membrane, so you can see where the basement membrane is, and from there if you move up you can see that there are many layers of nuclei, which tells you that this kind of epithelium is a stratified epithelium. In the ureter, what you see is a stellate lumen, because the ureter is a very, very muscular organ. It tends to throw the lumen into folds, and sometimes you'll see a very narrow lumen. In fact, in this image you are looking at a fairly good sized lumen. And then the arrow is now pointing to the smooth muscle in the top. Again, like I said, the ureter is a very muscular organ which has very thick smooth muscle layer. In the lower picture, in the lower image, which is that of the urinary bladder, since it's under high power, you can't see the muscle layer, but what you're looking at is the lamina propria, the connective tissue which is present just below the epithelium. Now here I have a little activity for you, and what you have here are two slides, slide 1 and slide 2. What I would like you to do for slide 1 is first identify what the arrow is pointing to. So identify whether it's a PCT, DCT or collecting duct, and then identify whether the image belongs to the cortex or the medulla. In the lower image, identify what structure the arrow is pointing to. Now I will pause for a minute, and after you're done identifying, I'd like you to hit the play button just below me, and then we can resume the video. Well, let's examine what your answers were. If you answered collecting duct for slide 1, then you are right, because you can see that it's light, the stain is very light, you can see a large lumen. And if you answered PCT, then you're wrong, because one, you do not see that fringed border, neither is it darkly stained. And again, for distal convoluted tubule, here the stain is too light and the structure is rather large to be a distal convoluted tubule. The arrow now points to a structure which is actually the thick segment of the loop of Henle, and that could have been mistaken for the distal convoluted tubule. But usually you will identify a distal convoluted tubule when you have a proximal convoluted tubule to compare it to. Now for the second slide, if you answered proximal convoluted tubule, you are right, and that is because you can see that fringed border, and it's not a collecting duct, and neither is it a distal convoluted tubule. In fact, the arrow now points to the distal convoluted tubule, and you can see the difference between this and the previous arrow, which was on the proximal convoluted tubule. Now for slide 1, you should have identified this as the medulla because it did not have the glomerulus, it did not have proximal and distal convoluted tubules. All that you can see here are collecting ducts and loops of Henle. And even if you thought they were distal convoluted tubule, the absence of a glomerulus and proximal convoluted tubule means that this is not the cortex. The lower image, however, would be one of the cortex. Let's go on to another activity where again we have two slides, slide 1 and slide 2. And I would like you to identify in slide 1 where the arrow is lying. Is it lying on the parietal layer of the Bowman's capsule? Is it lying on the visceral layer? Or is it in the capsular space? In slide 2, what I want you to identify is what is the arrow pointing to? Is it pointing to the basement membrane, the cuticle, or is it pointing to the layer of smooth muscle? And again, I'm going to pause for a second and wait for you to answer these two slides, and then we'll get back to them. And remember to hit the play button when you're done. Now for slide 1, if you chose capsular space, that is excellent, because that's exactly where the arrow was lying. The parietal layer is on the outside, the visceral layer is on the inside, and the arrow was slap in the middle of the capsular or Bowman space. Again, now you can see that this image is from the cortex of the kidney. In slide 2, again, if you chose the basement membrane, then you are correct, because the arrow was pointing to a line from where the epithelium started. So that is the basement membrane. It was not pointing to the cuticle, because the cuticle actually is in intimate contact with the lumen. And neither was it pointing to the layer of smooth muscle. So hopefully all of you identified slide 2 as the arrow in slide 2 as the arrow pointing to the basement membrane. Now that concludes this activity and this video, and I hope this was helpful to you. And as always, I'd like you to rewind, go through all the slides, listen and look carefully, and feel free to email me again if you have any suggestions or if there's anything that you did not understand.